So let's get with the topic amino acids. So basically you will learn how exactly is amino acids. Now let's split the word and see what exactly is amino acid. As you can see, has here I am mentioned that amino means basically there is a presence of amino group within the structure and acid group. So how exactly they look I had mentioned here, and you will see that how, see, this basic structure of amino acid is going to follow throughout the 20 essential amino acids we are going to cover in this video. Now how these amino acids, let us see whether how these amino acids are going to look. So there are around 20 amino acids available in nature, which is naturally occurring, we say naturally occurring amino acids, and likely there are 21st and 22nd. So what do we call these amino acids as? This 21st amino acid is called as cysteine, and the 22nd amino acid is called as pyroglycine. So this is how the amino acid is going to be look. So let's start with a very basic amino acid named as glycine. So how our glycine is going to look, let's see it. Okay, so what do you all think how our glycine is going to look? Just now I showed you a structure in which we draw a acid group which is very much near to a methyl, and then with one hydrogen is replaced by amino group. This is how our glycine is going to look around. Now you should remember that on the basis of this glycine structure we are going to connect with the next upcoming structure name as, in this video I am very much going to concern with the structures. Okay, so we'll make a alanine name amino acid from glycine. I will mention the source from glycine. So let's see what the alanine look like. Now listen, draw everything as given in the glycine, and there are some minute changes what we are going to make. Now what are the minute changes we are going to highlight? Now here we need to attach, so we had removed one hydrogen, we had replaced one hydrogen with methyl group. This is how we are going to draw alanine, okay, which is from glycine. Now we have to look on a very important classification from which we are going to derive the further amino acids. From now alanine we will derive phenylalanine, and from phenylalanine we will derive tyrosine. I hope so this is clear. Now there is one more amino acids we are going to derive from alanine, which is serine, and from serine we are going to draw threonine. And what the next is cysteine. There is one more amino acid we can derive in which E is not present, which is called as cysteine, which is not that as far as so important. After that we got methionine, which is derived from cysteine, and two amino acids which are directly drawn from the structure of alanine. Okay, so what they are? Tryptophan, and next is histidine. How many amino acids we had covered till now? Here is glycine 1, let me draw it with green one so you can cover 1, then alanine 2, phenylalanine 3, tyrosine 4, serine 5, 6 threonine. Wait a minute, there's a spelling mistake. So the E was missing. Okay, 6, 7, 8, 9 and 10. Out of 20 essential acids, we are going to cover the very initial 10 amino acid, which is phenylalanine. From that we are going to derive tyrosine. Then there is serine. From serine we are going to derive threonine. Then there is cysteine. From cysteine we are going to derive methionine. And the directly derived from alanine are tryptophan, histidine. So you should these two structures are little bit complicated, but they are easy. So let's get started with phenylalanine. So how phenylalanine is going to look? So here it is, the structure of phenylalanine. Okay, so from where we are going to derive it? From alanine. Now see how it is going to look. Draw alanine first of all. Whenever you have a confusion, let's start with alanine. Okay, CH2, this is our carboxylic group. You can either draw like this or open chain. Okay, and then this is amino group. Now what we have to do is, in this methyl you need to replace one hydrogen. You need to replace one hydrogen with phenyl group. How you are going to do? Let's look upon it. CH2, CH2, NH2. For your understanding, I am going to open this structure. So this is how it is going to look. Okay, so you just need to understand what changes we had made. So just highlight that changes so that we are not going to get confused. So this is a structure. This is the structure of phenylalanine. Okay, you should remember this one. Okay, now from this phenylalanine we are going to make tyrosine. So how we are going to make? Let's look upon it. Tyrosine. Now how we are going to draw tyrosine? From phenylalanine. From phenylalanine. Okay, from phenylalanine. So how we are going to draw? From phenylalanine. Now how it is going to look upon? Take this particular structure and draw it as it is. Okay, CH2, CH2, our amine group, and this is our acid group. What we have to do is, we have to just nothing but just add OH on para position to this. So what we are going to call it is a phenolic hydroxyl group. So I hope so these two amino acids are clear. So let's move on to the further amino acid, which is serine. So let me revise you all. Here we completed with phenylalanine and tyrosine. So let's go to serine and threonine. How this serine is going to derive? Let's say first of all. So I got some mnemonics for you all. Let's remember it as SIR, and it is going to derive from alanine. So how? Let's draw alanine first of all to understand what it is. Always remember, just keep one hydrogen here vacant to draw a structure, and else everything will be same, because it is amino acid. What it is amino acid. Okay, now here CH2 is going to, the one hydrogen of CH2 is going to replace by hydroxyl group. Now the question arise, how you are going to remember is. हिंदी में हम लोग बोलते हैं सर ने अल्कोहल पी. So how we are going to remember it as सर ने अल्कोहल पी. Just remember in this way. Just remember this way, so you are not going to forget it. Next one is you need to understand what is threonine. So we are going to derive threonine from serine. For this, just don't just take this. You should write the whole structure. I'm just copying for the longevity of this YouTube video. So you should draw a CH3 here. I hope so you got it, how this threonine is going to look upon. So for remember this, you can also remember like this. सर के साथ में बैठ कर 3 बॉटल अल्कोहल पी रहा था. So this is how you are going to remember this particular. May means methyl group. सार बैठ कर C. What you need to remember is सर मतलब 3 we are going to derive it from serine के साथ बैठकर में, this is methyl group, बैठकर 3 बॉटल, 3 means this threonine bottle alcohol पी रहा था, for remembrance of alcohol. So now we have again completed with two amino acids, serine, threonine. The next is cysteine and methionine. So how we are going to derive, let's talk about it. So here is your 7th amino acid called as cysteine. How cysteine, just remember cysteine में S है तो S से हम लोग will call it a sulfur containing amino acid. Nothing but we have to again draw with alanine, CH2 CH C double bond O OH. Here we got with amine, and here there's nothing but just draw with SH. Now for your understanding, let me clear you all this. What are the names of this group? Sulfur, we can call it as sulfur. We can also call it as mercapto. We can also call it as sulfahydryl. We can also call it as thio alcohol. These are some names of this cysteine. Now let's move on to the next amino acid, which is methionine. It is quite interesting, and for those who are very much interested in knowing cysteine, which has a disulfide bond and the whole structure is repeated, this one, okay, this will somewhere around यहाँ पे सल्फर होगा and the whole structure is going to repeat. So this is how this cysteine is going to look around. So for going further, we are again moving to the next amino acid, which is methionine. Now methionine will be derived from cysteine. Now let's draw cysteine. Before, you just need to remember cysteine, and from that you can remember all this further structure. CH2 and SH. Just remember, just add CH2. Okay, this is our alanine structure. Okay, now what the next thing we are going to do is, here add CH2 group and join this one with CH3. How? Let's see. This whole structure will be look like this. SH, this there is a presence of 2 CH2 groups and the rest structure is same. So this is how our methionine look like. Okay, so let's move on to the next amino acid, which is tryptophan. For this you should remember mnemonics: trip 3, okay, and fan. So what we are going to do is remember like this. We had planned a trip in Indore. This Indore will be remembered on behalf of indole ring. Now how this indole ring is going to look, let's take it, because we have to draw alanine. Draw it first. NH2 COOH. Now let's look upon how this indole ring, you just have to remember it on any case. So this is how indole ring look like. Here it is a 6 member. Okay, so how we are going to name it? Right from starting from the hetero, 1 2 3. Never take this fuse ring. Start with here 4 5 6 7. Let's fulfill the bonds. So always remember, in ESR, electrophilic substitution reaction, in indole ring we are going to substitute at third position. So we have attached this our alanine group on the third position. Then the next one is, and next and the very last in this 10 amino acids, which is histidine. Now how you are going to draw histidine? Again draw our beloved alanine, which looks like this. So nothing but add a imidazole ring. I hope so everybody know how to draw the imidazole. If you don't, just look upon how I am drawing. Okay, there's a presence of nitrogen on first and third position. Okay, here is NH. Okay, so always remember, you can either attach on 4th or 5th. Don't get confused whether you will see a alanine attached to 5th position. Both are going to represent the histidine. So nomenclature starts like this: 1 2 3 and 4 5. So here we complete with 10 amino acids which are essential. On later we are going to see the other 10 amino acids. Thank you.